The mystery of Rh-negative blood has fascinated scientists, historians, theologians, and curious minds for decades, not because it offers easy answers, but precisely because it raises questions that cut across biology, history, identity, and the limits of what we believe we know about human origins. To speak about Rh-negative blood is not merely to discuss a medical classification used in transfusions or pregnancy monitoring, but to open a window onto one of the most intriguing anomalies in human biology. An anomaly that resists simple explanations and continues to provoke debate in both scientific and cultural spheres. From the very beginning, the subject draws attention because it touches something deeply human. The question of where we come from, how we are connected, and why some of us carry biological traits that seem, at least on the surface, to stand apart from the rest of humanity. In modern medicine, the Rh factor is treated as a practical matter, a plus or minus sign attached to blood type that can mean the difference between compatibility and danger during transfusions or pregnancy. Yet behind this seemingly straightforward classification lies a biological puzzle that has never been fully resolved. The majority of humans on Earth are Rh positive, meaning they possess a specific protein, the D antigen, on the surface of their red blood cells. A smaller percentage, estimated at around 15% globally, but distributed unevenly across populations, lack this antigen and are classified as Rh negative. This absence, rather than the presence of something unusual, is what makes Rh negative blood so curious. Because from an evolutionary perspective, the question immediately arises: why would a significant portion of humanity lack a factor that appears to be dominant and widespread in the species? To understand why this question matters, one must first consider how genetics usually works in human populations. Traits that offer no disadvantage or that confer a benefit tend to spread over time, while traits that cause serious reproductive problems are usually selected against. The Rh negative factor, however, presents a paradox. When an Rh negative mother carries an Rh positive fetus, her immune system can, under certain conditions, recognize the fetus's blood as foreign and mount an immune response. Before modern medicine developed preventive treatments, this incompatibility could lead to serious complications, including miscarriage or the death of the newborn in subsequent pregnancies. From a purely evolutionary standpoint, this should have caused the Rh negative trait to diminish significantly over time, yet it has persisted, and in some populations it appears at unusually high frequencies. This persistence alone suggests that the story of Rh negative blood is more complex than a simple genetic accident. As researchers began to map the distribution of Rh negative blood around the world, further questions emerged. The trait is most common among populations of European descent, particularly in regions such as the Basque Country, between Spain and France, where the percentage of Rh negative individuals can reach as high as 30% or more. It is also found at notable levels among certain Celtic populations, and to a lesser extent in parts of North Africa and the Middle East. In contrast, Rh negative blood is relatively rare among East Asian populations and indigenous peoples of the Americas, where it appears only in small percentages. Such an uneven distribution is unusual for a trait that is supposed to be part of a shared human genetic heritage, and it has led many to wonder whether historical isolation, ancient migrations, or even unknown selective pressures played a role in shaping this pattern. The Basque case, in particular, has attracted intense interest because the Basques are already known for their genetic and linguistic uniqueness. Their language, Euskara, is unrelated to any other known language family, and genetic studies have shown that the Basques preserve markers associated with some of the earliest populations of modern humans in Europe. When the high prevalence of Rh negative blood among the Basques was documented, it added another layer to their enigmatic profile. Some researchers have suggested that the Rh negative factor could be a remnant of ancient European populations that existed before later migrations spread Rh positive genes more widely. Others argue that genetic drift, combined with long-term geographic and cultural isolation, could account for the concentration without invoking more exotic explanations. Yet even these hypotheses leave unanswered questions, particularly about why similar levels of Rh negativity are not found uniformly across all regions thought to be inhabited by ancient European populations. As the scientific debate continued, the mystery of Rh negative blood began to spill beyond academic journals into popular culture, where it took on a life of its own. Writers, documentary producers, and alternative theorists began to connect the trait to ancient myths, lost civilizations, and even extraterrestrial origins. While mainstream science remains skeptical of such claims, their popularity reveals something important about the topic. Rh negative blood resonates with a deeper human fascination with difference and origin, with the idea that some people might carry within their bodies a trace of something ancient, hidden, or exceptional. This fascination is not merely sensationalism. It reflects a genuine gap between what science can currently explain with certainty and what remains speculative. One of the most frequently cited arguments in these alternative narratives is the idea that Rh negative blood represents a deviation from the rest of the animal kingdom. It is often claimed that all other primates are Ra positive, implying that Re negative humans must have a different or unusual lineage. While this claim is sometimes oversimplified, it does highlight an interesting point. The Rh system, as we understand it, is particularly relevant to humans, and its evolutionary history is not entirely clear. The Rh factor is named after the rhesus monkey, in which a similar antigen was first studied, but the relationship between human Ra types and those found in other primates is more complex than popular accounts suggest. Nonetheless, the perception that Ra negative blood is somehow outside the norm has fueled speculation for decades. Another layer of intrigue comes from the reported correlations between Rh negative blood and certain physical or psychological traits, claims that range from the mildly curious to the outright controversial. Some have suggested that Rh negative individuals are more likely to have lighter eye colors, increased sensitivity to sunlight, or lower body temperatures, while others speak of heightened intuition or unusual psychological characteristics. From a scientific standpoint, most of these claims lack robust empirical support and are difficult to separate from cultural myths or confirmation bias. Yet the persistence of these ideas underscores how the Rh negative question has become intertwined with broader narratives about identity and difference, narratives that go beyond measurable biology into the realm of meaning. Despite the noise generated by speculation, the core scientific question remains unresolved. Where did the Rh negative factor originate and why has it persisted? Modern genetics tells us that the Rh negative trait is caused by mutations or deletions in the Rhd gene, resulting in the absence of the D antigen. Multiple variants of these mutations exist, suggesting that Rh negativity did not arise from a single event, but may have developed independently in different populations. This multiplicity complicates any attempt to trace the trait back to one origin or one historical moment. At the same time, the fact that Rh negative blood appears with such frequency in certain populations suggests that random mutation alone may not fully explain its distribution. Some researchers have proposed that the Rh negative factor could be linked to ancient selective advantages that are no longer obvious today. For example, it has been suggested that Rh negative individuals might have had some form of resistance to particular diseases or environmental conditions in the distant past. Such advantages could have balanced out the reproductive risks associated with Rh incompatibility, allowing the trait to persist. However, concrete evidence for specific advantages remains elusive, and without it, these ideas remain hypotheses rather than established facts. The role of modern medicine in shaping our understanding of Rh negative blood cannot be overstated. Before the development of Rh immunoglobulin treatments in the mid-20th century, Rh incompatibility was a significant cause of infant mortality in certain populations. The introduction of preventive medicine dramatically reduced these risks, effectively removing one of the strongest selective pressures against Rh negativity. This medical intervention, however, came very late in human history and cannot explain the long-term persistence of the trait. Instead, it highlights how the biological puzzle existed long before we had the tools to manage its consequences. What makes the mystery of Rh-negative blood particularly compelling for a long-form exploration is that it sits at the intersection of hard science and unanswered questions. On one side, we have genetics, immunology, and population studies, disciplines grounded in data and experimentation. On the other, we have history, anthropology, and myth, fields that deal with incomplete records and human interpretation. The Rh negative factor forces these domains into conversation with each other, because no single discipline can fully account for its story. This interdisciplinary tension is part of what keeps the topic alive and relevant, even as specific theories rise and fall. As we move deeper into the subject, it becomes important to maintain a clear distinction between what is known, what is plausible, and what remains speculative. The allure of dramatic explanations is strong, especially in an age where mystery sells and certainty can feel limiting. Yet the true fascination of Rh-negative blood lies not in definitive answers, but in the way it exposes the boundaries of our knowledge. It reminds us that even in a species as thoroughly studied as our own, there are still fundamental aspects of our biology that resist simple narratives. In the pages that follow, this exploration will continue to examine the origins, implications, and interpretations of Rh-negative blood with a critical but open-minded approach, rather than presenting the topic as a solved riddle or a sensational secret. It will treat it as an ongoing inquiry, one that reflects the complexity of human history itself. By looking at genetic evidence, historical patterns, and the cultural meanings that have grown around this trait, we can begin to understand not just why Rh-negative blood is mysterious, but why that mystery matters, and why it continues to capture attention in science, media, and the human imagination alike. As the discussion around Rh-negative blood moves beyond its basic biological definition, it becomes necessary to look more closely at the mechanisms of heredity and population genetics that govern how such a trait can appear, spread, and persist across thousands of years. At its core, the Rh factor is inherited according to Mendelian principles, where the Rh positive allele is dominant and the Rh negative allele is recessive, meaning that an individual must inherit 2 copies of the negative variant to express the Rh negative phenotype. This simple genetic rule, however, does not translate into a simple historical explanation, because dominance alone does not guarantee prevalence, nor does recessiveness imply rarity, especially when populations experience isolation, bottlenecks, migrations, and selective pressures that alter genetic frequencies over time. When early geneticists began examining the Rh system in detail, they quickly realized that the label Rh negative masked a surprising level of complexity. There is not a single Rh negative mutation, but rather multiple genetic configurations that result in the absence of the D antigen. In some populations, the Rhd gene is entirely deleted, while in others it is present but altered in such a way that the antigen is not expressed on the surface of red blood cells. This diversity suggests that Rh negativity did not arise from one unique ancestral event, but may have emerged independently in different human groups, or at least undergone significant modification as populations diverged. Such a pattern challenges any explanation that relies on a single point of origin and instead points toward a more intricate evolutionary history. Population studies have shown that genetic drift plays a powerful role in shaping the frequency of Rh-negative blood in small, relatively isolated communities. Chance alone can cause certain traits to become more common over generations, even if those traits offer no clear advantage. If a founding population happened to include a higher proportion of Rh-negative individuals, and if that population remained isolated through geography, culture, or social structures, the trait could reach unusually high frequencies without requiring any extraordinary explanation. This mechanism is often invoked to explain the Basque example, as well as similar patterns observed in island populations or culturally insular groups elsewhere in the world. Yet while genetic drift provides a plausible framework, it does not fully satisfy those who note that isolation alone does not always lead to such pronounced differences in other genetic traits. Another important factor to consider is migration, particularly the large-scale movements of human populations during the Paleolithic and Neolithic periods. As modern humans spread out of Africa and into Europe, Asia, and beyond, they encountered new environments, formed new communities, and sometimes interbred with other hominin groups. Each migration wave carried a subset of the genetic diversity of its source population, and each subsequent settlement reshaped that diversity through both adaptation and chance. The uneven distribution of Rh-negative blood may therefore reflect ancient migration routes, with certain lineages carrying higher frequencies of the trait and leaving a stronger genetic imprint in specific regions. Tracing these routes, however, is challenging because the genetic signals become increasingly blurred the further back in time one looks. The question of interbreeding with archaic humans, such as Neanderthals, has also entered the discussion, particularly as advances in ancient DNA analysis have confirmed that non-African populations carry small but significant amounts of Neanderthal genetic material. Some have speculated that Rh-negative blood could be linked to such archaic introgression, although current evidence does not support a direct connection between known Neanderthal genes and the Rh system. Still, the very fact that modern humans are not genetically isolated from other hominin species complicates any attempt to define a single, linear origin for traits like Rh negativity. Human evolution, as it turns out, is more like a braided river than a straight line, with branches that split, merge, and sometimes disappear entirely. Beyond genetics, the immunological aspect of the Rh factor introduces another layer of complexity. The immune response associated with Rh incompatibility is highly specific, involving the recognition of the D antigen as foreign when it enters the bloodstream of an Rh-negative individual. This specificity raises questions about how and why such a system evolved in the first place. The immune system is shaped by constant interaction with pathogens, parasites, and other threats, and it is possible that the Rh system is a byproduct of evolutionary pressures unrelated to reproduction. In this view, the reproductive complications associated with Rh incompatibility are an unintended consequence of an immune mechanism that evolved for other reasons, a reminder that evolution does not optimize for human comfort or convenience, but for survival in a changing environment. Historical records, while limited in their ability to address genetic questions directly, provide valuable context for understanding how Rh-negative blood may have influenced human societies before it was scientifically understood. In times when infant mortality was high and medical explanations were scarce, repeated pregnancy losses within certain families or communities could easily be attributed to supernatural causes, curses, or divine will. While it is speculative to link such beliefs directly to Rh incompatibility, it is not unreasonable to suggest that unexplained reproductive patterns may have contributed to cultural narratives about fate, bloodlines, and inheritance. Blood, after all, has always held powerful symbolic meaning in human cultures, often associated with identity, purity, and continuity. This symbolic weight is one reason why the discovery of the Rh factor in the 20th century had an impact that extended beyond medicine. It challenged long-held assumptions about blood compatibility and forced a re-evaluation of ideas that had previously been taken for granted. The realization that two people of the same ABO blood type could still face serious risks due to the Rh factor underscored the hidden complexity of human biology, a complexity that had been invisible until the tools of modern science made it measurable. For some, this discovery reinforced a sense of humility in the face of biological reality, while for others it opened the door to speculation about deeper meanings encoded in the blood. As the topic gained attention outside scientific circles, it became increasingly entangled with narratives about ancestry and identity. In some popular accounts, Rh negative blood has been portrayed as a marker of elite lineage, ancient nobility, or even spiritual distinction. These ideas often draw selectively on historical facts, such as the concentration of Rh negativity in certain European populations, while ignoring the broader global context and the absence of evidence linking the trait to social status or cultural achievement. From a scholarly perspective, such interpretations are problematic not only because they lack empirical support, but also because they risk reinforcing myths about biological hierarchy that have caused real harm in the past. At the same time, it would be a mistake to dismiss all cultural interpretations as mere fantasy. The ways in which people understand and interpret biological differences can have tangible effects on social behavior, self-perception, and group identity. For individuals who discover that they are Rh negative, the information can carry emotional weight, especially when encountered through narratives that frame the trait as rare or mysterious. This psychological dimension is part of the Rh negative phenomenon, shaping how the topic is discussed and remembered, even if it lies outside the strict boundaries of scientific explanation. In recent decades, advances in genomics have provided more precise tools for investigating the origins and implications of the Rh negative trait. Large-scale genetic studies have made it possible to compare Rh-related genes across populations with unprecedented resolution, revealing patterns that were previously invisible. These studies suggest that while Rh negativity is indeed more common in certain regions, it does not correspond neatly to any single ancestral group or historical event. Instead, it appears to be the result of multiple factors acting over long periods of time, including mutation, drift, migration, and selection, none of which alone can account for the full picture. This multifactorial perspective aligns with a broader shift in how science understands human variation. Rather than seeking simple explanations for complex traits, researchers increasingly emphasize the interplay of many small influences, each contributing incrementally to observed outcomes. In this framework, the mystery of Rh negative blood is not a sign of hidden knowledge waiting to be uncovered, but a reflection of the inherent complexity of biological systems. The unanswered questions persist not because of secrecy or suppression, but because the processes involved are diffuse, probabilistic, and deeply rooted in deep time. Yet even this sober scientific perspective leaves room for wonder. The fact that a small molecular difference on the surface of red blood cells can have such far-reaching implications, from pregnancy outcomes to cultural narratives, is itself remarkable. It illustrates how biology operates simultaneously, at microscopic and macroscopic scales, linking genes to lives and lives to histories. The Rh factor is a reminder that our bodies carry the legacy of countless generations, encoded not as stories or symbols, but as sequences of DNA shaped by forces we are only beginning to understand. As this exploration continues, the focus will shift toward examining the more controversial interpretations of Rh negative blood, not to endorse them uncritically, but to understand why they have gained traction and what they reveal about the human desire for meaning in the face of uncertainty. By situating these ideas within a broader scientific and historical context, it becomes possible to separate what can be reasonably considered from what belongs more to the realm of myth, while still appreciating the cultural significance of both. In doing so, the mystery of Rh negative blood emerges not as a single question with a single answer, but as a multifaceted inquiry into who we are, where we come from, and how we make sense of the differences that define us. As discussion turns toward the more controversial interpretations of Rh negative blood, it becomes clear that the persistence of these ideas cannot be explained solely by misunderstanding or misinformation, but rather by a convergence of historical gaps, scientific uncertainty, and a deeply rooted human inclination to search for exceptional origins. Whenever a biological trait appears unevenly distributed, poorly explained, or associated with tangible effects on reproduction and health, it becomes fertile ground for narratives that extend beyond conventional science. In the case of Rh-negative blood, this fertile ground has given rise to theories that range from speculative anthropology to outright cosmological conjecture, each attempting to answer questions that biology alone has not definitively resolved. One of the most frequently repeated claims in popular discourse is that Rh-negative blood represents evidence of a non-standard human lineage, sometimes framed as a remnant of an ancient advanced civilization, or even of non-human ancestry. These narratives often draw upon the absence of the D antigen as a symbolic marker, interpreting negative not merely as a medical classification, but as an absence that must itself signify something extraordinary. From a scientific perspective, absence is simply one of many possible outcomes of genetic variation. Yet culturally, absence tends to invite interpretation, particularly when it challenges expectations of uniformity within a species. This psychological tendency to read meaning into absence plays a significant role in how Rh-negative blood has been framed outside academic circles. Historical myths about divine bloodlines, chosen families, or semi-divine rulers have further fueled these interpretations. Across many cultures, blood has been seen not only as a biological substance, but as a carrier of essence, destiny, and legitimacy. Royal families often claimed descent from gods or legendary heroes, and blood purity was frequently invoked to justify social hierarchies. When modern audiences encounter information suggesting that certain blood traits are rarer or biologically distinct, it is perhaps unsurprising that these ancient symbolic frameworks resurface, recontextualized in the language of genetics. Rh-negative blood, in this sense, becomes a modern vessel for very old ideas about distinction and origin. Another recurring element in alternative theories is the association of Rh-negative blood with ancient texts and religious traditions. Some interpretations draw upon passages from sacred writings, reading them through a contemporary biological lens and suggesting that references to pure blood, giants, or sons of gods might encode forgotten genetic realities. While such readings are not supported by mainstream scholarship, they highlight an important dynamic. When scientific explanations feel incomplete or inaccessible, people often turn to symbolic systems that offer coherence and narrative closure. The appeal of these interpretations lies not in their evidentiary strength, but in their ability to weave disparate elements into a single, compelling story. It is also worth noting that the language used in discussions of Rh-negative blood often amplifies its perceived mystery. Terms like rare, ancient, and unexplained carry emotional weight, even when used accurately. In reality, Rh-negative blood is neither vanishingly rare nor entirely unexplained, but the remaining gaps in understanding create space for exaggeration. Media representations, particularly in documentaries and online platforms, sometimes emphasize these gaps while downplaying the substantial body of knowledge that already exists. This selective emphasis can unintentionally reinforce the idea that Rh-negative blood is a scientific anomaly bordering on the supernatural, rather than a complex but natural outcome of human genetic diversity. From a sociological perspective, belief in exceptional biological traits often correlates with periods of uncertainty or identity crisis. In times when traditional narratives about history, religion, or progress are questioned, biological markers can become anchors for alternative forms of meaning. Identifying as part of a rare or mysterious group offers a sense of belonging and distinction that may be lacking elsewhere. For some individuals, learning that they are Rh-negative becomes more than a medical fact. It becomes a personal symbol, a way of situating oneself within a broader, if speculative narrative about humanity's past and future. At the same time, there's a risk in allowing such narratives to go unchallenged, particularly when they veer into claims of superiority or exclusion. History provides ample examples of how misinterpretations of biology have been used to justify discrimination, oppression, and violence. While most contemporary discussions of Art negative blood do not reach such extremes, the underlying logic of exceptionalism warrants careful scrutiny. Scientific literacy plays a crucial role here, not in dismissing curiosity, but in providing tools to evaluate claims critically and distinguish between evidence-based conclusions and imaginative extrapolation. Returning to the scientific domain, recent research has increasingly focused on placing the Rh system within the broader context of human genetic variation, rather than treating it as an isolated phenomenon. When viewed alongside other polymorphisms, such as those affecting lactose tolerance, skin pigmentation, or immune responses, Rh negativity appears less singular and more consistent with known patterns of diversity. Many traits exhibit striking geographic variation, reflecting historical migrations, environmental pressures, and random drift. The difference with Rh negative blood is that its medical implications make it more visible, more consequential, and therefore more likely to attract attention. This visibility is further enhanced by the role of Rh compatibility in pregnancy, an area where biology intersects directly with deeply personal experiences. For couples who encounter Rh incompatibility, the issue is not abstract, but immediate and emotionally charged. The need for medical intervention, the awareness of potential risk, and the language of immune conflict all contribute to a sense that something fundamental is at stake. These experiences, shared through personal stories and online communities, add a human dimension to the topic that purely statistical discussions cannot capture. They also reinforce the perception that Rh negative blood is not just another genetic variant, but something that reaches into the core of life and reproduction. As genetic testing becomes more accessible, more people are learning about their blood type and genetic traits earlier in life. This democratization of information has both positive and negative consequences. On one hand, it empowers individuals to make informed medical decisions and understand their bodies more deeply. On the other, it can expose them to a flood of unfiltered interpretations and theories, some of which blur the line between science and speculation. In this environment, Rh negative blood often functions as a keyword that leads not only to medical guidance, but to entire ecosystems of content promising hidden truths and forgotten histories. From an academic standpoint, the challenge lies in communicating uncertainty without undermining trust. Scientists are trained to acknowledge what is not yet known, but in public discourse, uncertainty is often interpreted as weakness or concealment. When experts say that the full evolutionary history of the Rh system is still being investigated, this openness can be misread as an admission that anything is possible. Bridging this gap requires careful explanation of how scientific inquiry works, emphasizing that unanswered questions are an expected and productive part of research, not evidence of suppressed knowledge. The future of Rh negative research will likely depend on advances in comparative genomics and ancient DNA analysis. As more ancient human remains are sequenced, it may become possible to track the frequency of Rh-related genes across different time periods and regions with greater precision. Such data could clarify whether Rh negativity increased, decreased, or remained stable in response to specific historical events, such as climate shifts or population replacements. While these findings may not satisfy every speculative narrative, they have the potential to ground the discussion more firmly in empirical evidence. At a deeper level, the enduring fascination with Rh negative blood reveals something about the relationship between science and meaning. Science excels at describing mechanisms, probabilities, and patterns, but it does not inherently provide narratives of purpose or identity. When people encounter a biological difference that affects their lives, they naturally seek stories that help them understand what that difference means, not just how it works. Rh negative blood, situated at the crossroads of biology, reproduction, history, and symbolism invite such storytelling in a way few other traits do. As this examination approaches its final stage, the focus shifts toward integration rather than resolution. The goal is not to eliminate mystery entirely, but to situate it appropriately, recognizing which questions are open, which are answered, and which belong more to the realm of cultural imagination than to biological fact. In doing so, Arch negative blood can be appreciated not as proof of hidden origins or cosmic intervention, but as a reminder of the layered complexity of human existence, where genes, histories, and meanings intersect in ways that resist simplification. As this exploration of R-negative blood reaches its conclusion, it becomes increasingly clear that the true significance of this subject does not lie in any single theory, claim, or unresolved detail, but in what the mystery itself reveals about the nature of human knowledge and the way we relate to our own biology. R-negative blood, stripped of sensational framing and examined in its full scientific, historical, and cultural context, stands as a reminder that even within a species as intensively studied as ours, there remain features that resist complete explanation, not because they are supernatural or forbidden, but because they are the result of long, layered processes unfolding across deep time. What modern science offers above all is not a final answer, but a framework for understanding. Genetics explains how Rh negative blood is inherited. Immunology clarifies why incompatibility occurs, and population studies show how uneven distributions can arise through migration, isolation, and chance. These explanations are powerful, yet they remain probabilistic rather than absolute, painting a picture of human variation that is dynamic rather than fixed. In this picture, Rh negative blood is neither an anomaly nor a marker of exceptionality, but one expression of the immense diversity that characterizes our species, shaped by forces that operate beyond individual intention or awareness. At the same time, it would be misleading to suggest that the scientific account completely dissolves the sense of mystery surrounding the Rh factor. The absence of a single, clean origin story, the persistence of the trait despite historical reproductive risks, and its striking concentration in certain populations all leave room for questions that are still actively investigated. These open questions are not failures of science, but invitations to further inquiry. And they remind us that understanding is always provisional, always subject to refinement as new evidence emerges. In this sense, the mystery of Rh negative blood is not a problem to be solved once and for all, but a case study in how knowledge evolves. Equally important is the recognition that human beings do not engage with biology in a vacuum. Blood has never been merely a physiological substance. It has always carried symbolic weight, shaping how people think about kinship, inheritance, belonging, and difference. When modern discoveries intersect with these older symbolic frameworks, the result is often a hybrid discourse where scientific facts coexist with mythic interpretations and personal meaning. R-negative blood occupies precisely this intersection, functioning simultaneously as a medical classification, a genetic trait, and a canvas onto which broader questions of identity and origin are projected. The persistence of alternative theories, from ancient lineages to non-human ancestry, should therefore be understood not only as a response to gaps in scientific explanation, but as an expression of a deeper human impulse. Faced with uncertainty, people seek narratives that provide coherence, significance, and a sense of place within a vast and often impersonal universe. For some, the idea that their blood connects them to an ancient or extraordinary past offers comfort or distinction. For others, it simply satisfies curiosity. A critical approach does not require dismissing these impulses outright, but rather situating them within a broader understanding of how meaning is constructed and why certain stories endure. From a medical and ethical perspective, the Rh factor also illustrates how scientific knowledge directly affects human lives. The development of treatments to prevent Rh incompatibility complications transformed countless pregnancies from high risk to routine, demonstrating how abstract genetic insights can lead to tangible improvements in well-being. This progress underscores the importance of grounding discussions of Rh-negative blood in accurate information, particularly when misinformation can cause unnecessary anxiety or reinforce unfounded beliefs. At the same time, it highlights how recent these advances are in the context of human history, reminding us that for most of our past, the biological realities of Rh incompatibility played out without explanation or intervention. Looking forward, ongoing research promises to deepen our understanding of the Rh system, but it is unlikely to deliver the kind of dramatic revelation that some narratives anticipate. More detailed genetic mapping, larger population data sets, and improved models of human migration will probably refine existing explanations rather than overturn them. This incremental progress may feel unsatisfying to those seeking definitive answers, yet it reflects the way science actually advances, through accumulation and correction rather than sudden disclosure. In embracing this process, we gain not only knowledge, but a more realistic appreciation of complexity. Ultimately, the story of Rh-negative blood is less about uncovering a hidden secret and more about learning how to live with unanswered questions. It invites us to reflect on the limits of certainty, the interplay between biology and meaning, and the ways in which scientific facts become woven into personal and cultural narratives. By approaching the topic with curiosity tempered by critical thinking, it becomes possible to appreciate both what is known and what remains open, without collapsing one into the other. In the end, Rh-negative blood does not separate humanity into categories of the ordinary and the extraordinary, but reminds us that variation itself is ordinary. The difference is built into the fabric of life. Each genetic trait, whether common or rare, carries the imprint of countless lives before us, of migrations taken and paths not taken, of survival and chance intertwined. To understand this is not to demystify the human story, but to deepen it, recognizing that our origins are complex, shared, and still in many ways unfolding. If this exploration leaves a lasting impression, it is not that Rh-negative blood holds the key to a forgotten past, but that the human body itself is a living archive, one that speaks in probabilities rather than certainties, and whose messages require patience, humility, and context to interpret. In listening carefully to those messages, we do not find a single answer to who we are, but a richer appreciation of how much there still is to learn, and why that learning matters.